Between the 17th and 19th centuries, the concept of xenotransplantation floated around the medical world through the distinguished actions of blood transfusions in patients experiencing pathological conditions. To understand the concept of xenotransplantation, we must first uncover the terminology given to such a procedure. In simple recognition, xenotransplantation involves grafting and transplanting organs and tissues amongst two different types of species, most commonly animals to humans. Whilst it sounds to be a simple concept of taking an animal's organs and implanting them into a human body, the steps needed to be performed to ensure a safe and successful surgery is the process of what occurs beforehand, which is the need to genetically modify animals' dsNA cells by taking away and adding to correctly allow the organ to work and not cause risks and organ failure to the human body. Let's take a look at how we can prevent these dangers from occurring.
The best strategy to minimize the risk of rejection during the pig-to-human heart transplant was to genetically modify the pig heart to be as closely replicated to the human heart as possible, and this was achieved through modifying the pig's DNA. DNA is known as the blueprint of life, as it provides cells with the information needed to function and display traits that make each individual unique. Genes are the various sections of DNA that control different functions and characteristics of human cells. Everything from eye color to heart function is influenced by the genes we possess. Although each individual's DNA is unique, there are significant similarities in genes for those part of the same species. Therefore, in order to close the gap between the differences between a pig and a human heart, Griffith and Mohiad found that altering the DNA of the pig heart to eliminate genes distinct for pigs and include human genes related to heart function would reduce the chance of rejection. And they were able to do this using a gene editing technology called CRISPR-Cas9. CRISPR-Cas9 allows us to make cuts in the human DNA at sites that contain genes that we want to eliminate. So the human body has natural cellular repair mechanisms to respond to any damage in the DNA. So when the cell senses that these cuts have been made in the DNA, it uses this repair mechanism to correct it. Normally, the cell will simply repair the DNA by adding back the genes that have been removed. However, in CRISPR-Cas9, scientists will actually add templates for new genes. So the cell will instead repair the DNA by incorporating these new desired genes in it. And now we have altered the human DNA. Griffith and Mohiad used the CRISPR-9 technology to remove 3 genes responsible for creating sugars found in pigs, but not humans, as the production of these sugars in the human body would likely trigger an immune response since they are foreign to the human recipient. A growth hormone gene was also knocked out from the pig heart to ensure the heart maintained a normal human size. And lastly, 6 genes involved in controlling inflammation in humans was added to the pig heart to allow it to mimic the human immune response. On January 7, 2022, Dr. Griffith and Dr. Mohiad removed the pig heart from the adult donor pig after altering its genes during its embryonic growth. The heart was stored in a box engineered to keep it vital until it was transplanted into Bennett's body that same day. It has now been over 2 months since the surgery, and Bennett is still in the recovery stage at the University of Maryland's hospital. Early on in his recovery, he did experience an abdominal infection due to the use of strong immunosuppressants to limit an immune attack on the new hearts. However, he later recovered from this infection. Also during his recovery, it was found that Bennett's new heart was beating with excessive force and speed, which led to a prescription of medication to help relax its activity. As of now, this pig-to-human transplant is viewed as a success. However, the full extent of its results will not be fully understood until we learn more about Bennett's condition in the future.
Transplantation is of particular importance because there's a huge lack of human donors, so other animals serve as a great substitute to save the lives of patients with organ failure. The concept of xenotransplantation are great candidates for xenotransplantation nowadays due to the fast maturation of organs and similar physiology to the human anatomy.
In choosing the best candidate animal for xenotransplantation, researchers often narrow it down to pigs and primates. Some reasons that pigs are preferred over primates include the fact that pigs can be bred intensively with large litters, meaning the number of available organs could be increased rapidly. Keeping pigs in sterile conditions is relatively easy. Pigs do not resemble humans the same way primates do, making people feel less guilt when raising pigs for transplantation. And the use of pigs is considered by some as acceptable because of their production for food. While it does seem strange that the organs of a different animal could function properly in a new host, a 98% DNA match between humans and pigs leads to pig hearts having a very similar size, anatomy, and function to human hearts, and they are quite compatible. Both hearts contain 4 chambers, consisting of 2 ventricles and 2 atria. There are also 4 valves that are consistent with the valves in the human heart, and the aorta is in a similar position. These similarities allow for blood to flow through a pig heart the same way it flows through a human heart. There are also some key differences. For one, the shape of the pig heart is a typical Valentine heart shape, while the human heart is more of a trapezoidal shape. Pig hearts also tend to be larger than human hearts. Pigs are 4-legged animals, and the heart is used to pumping horizontally, whereas in humans who stand upright, the heart pumps vertically. Additionally, in humans, the left atrium receives 4 pulmonary veins, whereas in pigs it receives only 2 pulmonary veins.
To deal with. The biggest concern post-surgery is rejection of the transplanted organ and increased susceptibility to secondary infections. Rejection is usually the primary concern following transplantation. The fear of rejection is even more amplified if the transplanted organ is from a non-human donor. Our body has curated mechanisms to prevent our immune system from attacking our own organs, tissues, and cells. However, any foreign material that enters our body is instantly eliminated by our immune system. Due to this process, an organ that comes from a different species is usually rejected within a few minutes in a process called hyperacute rejection. This is a process in which immune cells in the recipient's bloodstream attack the transplanted organ and eliminate it. The transplanted organ usually has antigens on its surface, which are like flags stating, "Hey, I am a pig heart, not a human heart," that signal the immune cells to trigger a defensive immune response, causing the organ to stop working. To bypass this mechanism of rejection, anti-rejection drugs, also known as immunosuppressive drugs, are administered to the patient. These drugs suppress the recipient's immune system, preventing it from being able to mount a strong response against the transplanted organ. This risk is the risk of infections. Because patients take immunosuppressive drugs, their immune system is weakened, making it unable to protect the body against foreign pathogens. These immunosuppressives can lead to a significant decrease in white blood cells, increasing susceptibility to many bacterial infections, making them more serious than they otherwise would be. Patients will have to adhere to strict medical regimens like taking multiple immunosuppressive drugs and having regular doctor visits, which include sampling or biopsies of the transplanted heart tissue to check for rejection, which can be an anxiety-inducing process. The transplantation process is very energy taxing, and it can take several months for a person to gain their energy back.
Many argue if it is right to harvest animals for their organs to save human lives. We do not have consent from these animals to do such a thing, and the unnatural living conditions which they are raised in is questionable. Animal rights movements also argue that raising pigs just to put their organs into humans is horrifying and inhumane. Another point is that how would you go about deciding who deserves the organ first? For example, a child who desperately needs an organ as soon as possible versus a mother of 5 who has been on the waiting list for a regular organ for a long time. In this case, how do we allocate the pig organs accordingly? Since the process is so new, would we create a waiting list or start giving them to those who need it most? What is fair and what is not?
In this video, we have discussed xenotransplantation, looking at the compatibility between human and pig hearts, post-transplant risk factors, and ethical concerns surrounding xenotransplantation. With many cardiovascular, kidney, liver, and lung diseases on the rise, coupled with the lack of people signing up as organ donors, there is a global organ shortage crisis. A potential solution that can improve and save lives is xenotransplantation. There is still a lot more research to be done, but with the first pig-to-human heart transplant completed, we are on our way there.